Good day everyone, I'm Zeejey Banag from STEM 11 LINNAEUS, welcoming you to the world of biotechnology.
What is genetic engineering?
Genetic engineering is the artificial manipulation, modification, and recombination of DNA or other nucleic acid molecules in order to modify an organism or population of organisms. The term genetic engineering is generally used to refer to the methods of recombinant DNA technology which emerged from basic research in microbial genetics. The techniques employed in genetic engineering have led to the production of medically important products, including human insulin, human growth hormones, and hepatitis B vaccine, COVID-19 vaccines, as well as the development of genetically modified organisms.
What is GMO?
Genetically modified organisms are organisms whose genome has been engineered in the laboratory in order to favor the expression of desired physiological traits or the generation of desired biological products in conventional livestock production, crop farming, and even pet breeding. It has long been the practice to breed select individuals of a species in order to produce offspring that have the desirable traits or that can be helpful to our present years. Genetically modified organisms are produced using scientific methods that include reproductive cloning and recombinant DNA technology, which I am going to discuss for this video. Recombinant DNA or rDNA is a technology that uses enzymes to cut and paste together DNA sequence of interest. The recombined DNA sequence can be placed into vehicles called vectors that ferry the DNA into suitable host cell where it can be copied or expressed. Now, let's proceed to the steps of recombinant DNA. The first step is isolation of DNA.
Being a nucleic acid enclosed within the nucleus, isolation of DNA is not an easy task. Isolation of DNA is an enzymatically controlled process where the plant or animal cells are treated with certain enzymes. Enzymes such as cellulase for plant cells, lysozyme for bacteria, and chitinase for fungi are used to isolate pure DNA from the cells.
Next is restriction enzyme digestion.
The isolated and purified DNA is treated with restriction endonucleases which cut the DNA into fragments. These restriction enzymes utilized in recombinant DNA technology are significant to detect the location at which the desired gene is introduced into the vector genome. The restriction endonucleases are sequence-specific, typically palindrome sequences, and snip the DNA at specific points. They inspect the length of DNA and trims it at a particular sites known as the restriction site. The desired genes and the vectors are snipped by the same restriction enzymes to acquire the complementary sticky ends. This ensures the task of ligases for binding the required gene to the vector is easier.
Next is amplification of gene of interest.
Polymerase chain reaction or PCR is a process to amplify the gene once the proper gene of interest has been cut using the restriction enzymes. Through this process, multiple copies of the gene of the interest can be produced. PCR proceeds in three stages: denaturation, annealing, and extension.
The next step is ligation of DNA molecules.
The purified DNA and the vector of interest are cut with the same restriction enzyme. This gives us the cut fragment of DNA and the cut vector that is now open. The process of joining these two pieces together using enzyme is DNA ligase is ligation. The resulting DNA is recombinant DNA.
The next and final step is the insertion of recombinant DNA into the host.
The host is the final tool of our DNA technology, which consumes the vector engineered with the desired DNA with the aid of the enzymes. This process is transformation. Bacterial cells do not accept foreign DNA easily. Therefore, they are treated to make them competent to accept new DNA. Insertion of the desired recombinant DNA into the host organism can be achieved in various ways. This includes biolistics or the gene gun, microinjection, alternate heating and cooling, usage of calcium ions, etc. The successfully transformed cells or the entities pass the recombinant gene to the offspring. So, that's it. That is the process of recombinant DNA.
Scientists have used recombinant DNA technologies to create many different types of recombinant animals, both for scientific study and for the profitable manufacturing of human proteins. For instance, mice, goats, and cows have all been engineered to create medically valuable proteins in their milk. Moreover, hormones that were once isolated only in small amounts from human cadavers can now be mass-produced by genetically engineered cells. In fact, the entire biotechnology industry is based upon the ability to add new genes to cells, plants, and animals. As scientists discover important new proteins and genes, these technologies will continue to form the foundation of future generations of discoveries and medical advances. Thank you for listening. Have a good day.